Good afternoon, everybody. Thanks for the introduction. I'll get right onto it because I've got three things to cover. Um, I'll talk about two of the projects that I'm involved with in terms of the breeding program and a discovery that I made a couple of years ago. And also the organizers have asked me to just talk briefly about fungicide use, especially with recent releases or recent registrations. I will talk about Neopestalotiopsis tomorrow. So if you guys are in the Queensland Berries tour, I'll talk more about that because I know that there's been some interest, particularly in the Queensland production with Neopestalotiopsis. So Australian strawberry breeding program. We do know that crown rot diseases are significant in our production. We've had reports of over 50%, I've seen 90% in our Queensland summer production. And I will probably draw some of the few parallels to Scott's presentations yesterday. But we do know that symptoms typically will involve gradual wilt or total plant collapse. And when you hack into that crown, you will see internal discoloration within the crowns and in those vascular tissues. Soil fumigation, fungicide applications are typically our methods of control that we do rely on. And as we heard from Scott, it's not, it doesn't fully eradicate some of the pathogens. And we can do a coordinated approach with different practices, but sometimes some of those may not be viable or may not be possible. And to exacerbate that, there are no resistant varieties at the moment with a number of these soil pathogens. And so the number one, or one of the main objectives of the research on our breeding program is that we're trying to put that resistance breeding up there. So this is the team. You guys would have met Jody Neal, there, and you would have seen her doing a lot of promotions during this conference. We are spread out as a national breeding program. We do have staff down at Wandin in Victoria and also in Western Australia. Okay. So just for those of you who don't know, in terms of our production, we do grow under 3 different climatic zones or geography. And essentially, each of those places have their own main crown rot issues. So in the Mediterranean, in Western Australia, we do have Fusarium as our main issue. In the subtropical, here in Queensland and up at Bundaberg, Cholecalciferum is our main pathogen. However, Macrophamina, as Scott alluded to yesterday, is pretty much the most significant out of all 3. Apart from dominating in the temperate region, we do see them in Western Australia and here in Queensland as well. I'll press that. And so, as a plant pathologist, I do all the culturing of pathogens. I guess you can call me the fun guy of the group. I prepare the inoculum, challenge the plants, and obviously over time try and assess them and in terms of their response to the pathogens. I'll skip through the methods and go straight into our collated results. So on this end for Fusarium wilt, Kabbalah is our susceptible control. And I guess everything on this left-hand side will have similar susceptibility to Fusarium. However, up on that end is where most of our accessions with some tolerance or resistance to Fusarium wilt. And I've highlighted some of the more common varieties and some of the varieties that we have developed in the breeding program. Similarly for Cholecalciferum, Camorosa is our susceptible control. And again, you can see there's probably 1 or 2 breeding accessions that are more susceptible than Camorosa. And again, up on this end is probably, displaying more resistance to Colletotricum. And again, we've had some recent releases in Sun Drench and obviously Red Rhapsody that shows some tolerance to Colletotricum. And for charcoal rot, and this is, I wanted to highlight, Albion is the major cultivar that is grown in the temperate region. And you can see why this work is so important. Because, I think 95% or 90% is that correct, Scott? Of Albion is used in the temperate regions. But, in our recent releases in Tali and Tamara, they are showing a good degree of tolerance to charcoal rot. And charcoal rot is also very close to me. I've recently completed a master's on it in terms of looking at sources of resistance and also our initial methods of inoculating for charcoal rot. So if you want to do some light reading, happy for you to Google. Powdery mildew, we also have started powdery mildew screening over the last couple of years. So I won't go through that now, but we do have it in our protected cropping and up in our tabletops, where we obviously can rely on natural inoculum of powdery mildew. And so this is just for the fruit. So you can see, I've got a picture of our more commonly used variety in Red Rhapsody here in the winter production. So you can see that it's roughly about there, comparing that with one of our breeding accessions that's got a very high tolerance to powdery mildew. And for guys that are in the temperate zone, Tali is obviously one of the newer ones, so hopefully, we are seeing some of that traits, in the temperate region. Now, you probably saw the phenomenal name. I think Andre asked yesterday about, we're breeding cultivars that are in the methyl bromide, that have been bred in the methyl bromide here. This is actually a very old variety, and we do have the germplasm in our collection. So you probably saw all the resistance levels of it were pretty much down there. So we are using this germplasm in our pre-breeding activities. So essentially, using it as parents, and some of the varieties we do have now do have the phenomenal germplasm in them. And so just to finish that up, we do have the accessions with resistance, and we are currently using them now in our breeding program. And we're also going to be using, and Katie, our other breeder, will also be looking into the genomic side of things to obviously take our breeding program to another level. Okay. Next one. This is the one I'm going to be very excited about. This is going to be the first time I've introduced this to the public, essentially. And again, I'll draw on Scotty's introduction on macrophamina yesterday, but also I'll just highlight again that it's, the current fumigants we have do not control or eradicate it fully. There has been national level research and state level research in terms of managing the pathogen, but obviously, having a coordinated approach can minimize that disease. However, even the best treatments of, the TIF plastic and the fumigation, you can still get about 10% plant losses due to macrophamina. A couple of years ago, looking through, accidentally I guess, looking through some of my plates, I just happened to stumble on an isolate, I guess. I can say that it's the genus Trichoderma genus, and I saw it kind of like dominating in some of the plates that also had macrophamina. And there's nothing. It's nothing new. We all know about Trichoderma and Bacillus, being aiding disease control. However, for strawberry and charcoal rot, there hasn't really been much done or anything done, not just in Australia, but internationally. So AG22 is the isolate, and according to the guys at the herbarium, this is the first known record of this species in their records and in Australia. I did look through the herbarium records for New Zealand, and to the best of my knowledge, I couldn't find it. There are no products derived from these species, and so I tried to, well, I did talk to my management, and they're happy to support. They did support me in terms of doing a small-scale glasshouse work, and I'll talk about them now. So that's him, or that's her, that's it. So in terms of dual plate confrontation, it did outcompete macrofamina significantly in a number of plates, and that's side by side, obviously. And I'm just thinking on the soil profile, if you have macrofamina on one side, and if you do surround that with the antagonist, what would happen? Perhaps something like that, where you just keep macrofamina in its little space, and the antagonist obviously overgrowing, the pathogen. And so from that, obviously I got excited. Let's do some work on pot plants. And so the first assay that I did was by using the isolate, doing a spore concentration, drenching the potting mix, and then 24 hours later, drenching it with macrofamina pathogen. And you can see 3 months, there is obviously a difference there in terms of plant losses from the macrofamina, and with the treated plants, obviously only very small losses. The next assay was dipping new transplant roots into the spore suspension of the isolate and planting that in pathogen-infected soil. And for the untreated, I just dipped it onto sterile water and put them into infected soil. And you can see that at 9 weeks, 80% against 10%, I think is pretty significant. It did eventually almost caught up, but the 40% deaths only really happened in the last couple of weeks of that trial period. And I guess you can probably think of it as, because these are spores in the soil, it probably doesn't last as long if it was obviously colonized the roots, and perhaps the macrofamina has actually increased itself, and then obviously have killed the plants, or most of the plants. But obviously, in terms of subsequent trial, I had a little bit more pot plants to use in the next one. But instead of just doing one treatment or one application, I did subsequent drenches as well, not just from dipping it at the beginning and doing more applications. So you can see by 16 weeks, we've almost got most of the plants dead. But if you do have 2, 3, or 4 more drenches, it's obviously provided some significant protection. So, implications. Isolate AG22 seems to have the potential to reduce incidence of macrofamina flagellina. This might actually fill the gap, because for those of you who fumigate, you say in temperate, where you fumigate in March, April, and then there's nothing else in terms of treatment in summer, this could be the product that could fill that gap to give that protection before the weather heats up or warms up. Obviously, for those who don't want to choose to fumigate, this could be a potential product, and those that are in the organic production could also be a potential product. It's obviously a softer, from a health perspective for users and for the environment, and hence, a future sustainable option. Currently, again, I have had support from my organization, and I am now, we do know that it could be a different thing when you're doing it in the lab, in a potted plant in the glasshouse. So I am currently doing some field trials now to evaluate the isolate in that sort of setting. And just to finish off this one, yes, again, I'm introducing it to everybody. There is an opportunity to work with us, with DAF, in terms of trying to see if we can further develop this isolate in a pre-commercial product. Again, it's a unique species, and there's no other products derived from the isolate. The potential is, it's not just strawberry. There's a lot of broadacre crops, horticultural crops affected by macrophyllina, and so perhaps this could be a thank you, and if there's any HIA or Hort Innovation people out there, it potentially could be a multi-industry project, and obviously would like to discuss more. I'd just like to acknowledge my technical assistant Tani, and also again DAF for supporting this work. And lastly, fungicide resistance management. I'll just go quickly about, you guys probably know what fungicides do, and again we do need the fungicides, mainly because we are a unique crop. We're not like other crops where there's a defined flowering, immature fruit, harvest, and then there's nothing else that happens. We are always constantly picking fruit. We're creating wounds all the time, and obviously, depending on the weather as well, that can support disease or development. So we do need that constant protection, ongoing protection to manage infections from pathogens. And so you guys would have heard of lunar sensation coming on just a few weeks ago. That we've also had intervene come on as well, which is really good in terms of having a different mode of action. But again, I do like having new products, but we do have to be careful if they are in the same group, because you guys will know, and I'm not saying anything new here, the label will tell you any of the same groups, you really just got to treat it as one. And my friend at Syngenta will confirm that for me. So essentially, for botrytis, group 7s, again, treated as one. There's a group 9 as well. For powdery mildew, all those group 7s, you do have to treat them as one. But then you also got to look into the core formulation. Again, the 12s in Moravis Prime and Switch, and in the Luna and Flint are also in the same group. So apart from that complexity, you also, and hopefully your agronomists are on top of it, but obviously depending on what you're trying to target, you also got to try and see if you can coordinate that during the year. So essentially, in terms of trying to avoid fungicide resistance, only use the registered products and always follow the label. Coordinate all those practices that you can, hygiene, regular monitoring, if you do have to spray, and again, optimize your spray equipment as well to make sure that you are putting them on nicely and properly, and the coverage is good. I do know that products will say, you can use it 2 in a row, but I'll just say, just keep rotating different products all the time, and limit the use of the more heavier or single-site fungicides. And if you can use those multi-site more often, that'll be better. Thank you. And from a whole industry perspective, Scotty and I did some work managing powdery mildew and trying to coordinate different groups used in the nursery. I think we should continue to keep doing that to make sure that we have a coordinated approach so that the products we're using in the nursery aren't being used. And if you heard Natalia's presentation yesterday and all the problems they have had with fungicide resistance, it's because their runners are coming from anywhere and everywhere, and they do not have control of what gets sprayed. We are lucky in Australia where we have a closed system. At least I would like to know what's being sprayed in the nursery. And if we can obviously coordinate different products at different levels. And this could also apply for other berries in their other nurseries as well. And if we could do that, that'd be good. So I'll finish off on that note. Thank you for your attention. And if there's any questions, I don't think we have, but all right. Oh, we do? All right, thank you. 30 seconds.
Yes.
Thank you.
Perfect timing. And we do have time for some questions for this one. Yep, we've got Lachlan down the front there.
Hey, Lock.
I don't know if you need it or not.
Oh, I should be right.
Yeah.
Sorry, Paul, just with the new products that have come out with the joint groups, do you recommend growers use those or stick to the traditional single-use products that are already on the market?
I won't say what you can use and what you shouldn't use, but it's really just managing. You may have a preference for one or the other products, but just obviously manage, especially those co-formulated ones, because once you use that other product, once you use that product with those two ingredients, then it probably takes out your other ingredient for other pathogen control. So yeah, thanks a lot.
I've been told to use the microphone, so we better pass
Oh,
that around.
okay.
There's another question over there, was there?
What?
Anyone on the spot? No questions about that new pathogen? It's good that you've got something to control it so quickly. No?
Okay, cool.
Excellent. Thank you very much,
you.
Apollo.
Cheers.
Excellent.